Hi students, today we are going to discuss about one more important short notes from carbohydrate metabolism, that is minor pathways. Okay, the important short notes is uronic acid pathway or glucuronic acid metabolism, right? Both are same. Uronic acid pathway or glucuronic acid metabolism. Usually they are asking the significance of the uronic acid pathway only or functions of the uronic acid pathway. Mostly they don't want to go for they are not asking to draw the pathway of uronic acid, but we should know about some 2-3 points about the metabolism also, right? So the first point is the initial products are glucose, which will be converted into glucose 6-phosphate, then 1-phosphate, finally UDP glucose formation. These four are the primary steps involved in uronic acid pathway. That is, glucose is converted into glucose 6-phosphate, glucose 6-phosphate converted into glucose 1-phosphate, then this glucose 1-phosphate combined one molecule of UTP, that is uridine triphosphate, and synthesis UDP glucose. So your first point will be formation of UDP glucose. Okay, this UDP glucose go for the first dehydrogenase reaction with the help of UDP glucose dehydrogenase. Usually dehydrogenase means they will be utilizing one NAD, which is a coenzyme for niacin, converted into NADH+. This UDP glucuronate is converted first into D form of gulonate, then which will be converted into L form of gulonate. So your second point is UDP glucuronate, third is L-Gulonate. So first you have to draw these three molecules in the uronic acid pathway. First one is these four, second one is UDP glucuronate, third one is L-Gulonate, right? After this L-Gulonate is converted into L-xylulose. I think you know the meaning of L and D. It is form of L form and D form, dextro form and levo forms of biochemistry. This L-xylulose, that is L-Gulonate, is converted into L-xylulose with the help of L-Gulonate decarboxylase. Decarboxylase. Okay. So this L-xylulose again converted into xylitol, xylulose and D-xylulose 5-phosphate. Right? You don't worry about the name. And the reactions here reductase, here dehydrogenase, here kinase enzyme involved. That is, L-xylulose is converted into dextro X, that is xylulose. X means this xylulose, D-xylulose 5-phosphate. Okay. This D-xylulose 5-phosphate finally enters into the pentose phosphate pathway. Now what is the significance or what is the relation between these two? Uronic acid end product is D-xylulose 5-phosphate, which will be entering into the pentose phosphate pathway. You know the significance of HMP shunt pathway. Finally it will be converted into glucose, right? Actually the initial product is glucose. Final product is also glucose. In between we are having so many compounds, right? After drawing these two pathways, two sections, we have one more section, that is L-Gulonate is converted into L-Gulonolactone. This compound, you should know the name of this compound, L-Gulonolactone, because this L-Gulonolactone only converted into L-ascorbate, which will be synthesizing ascorbic acid. For this the enzyme name is L-Gulonooxidase. L-Gulonooxidase. See the reaction. Here the product is L-Gulonolactone, which is converted into L-ascorbate, finally converted into ascorbic acid with the help of the enzyme L-Gulonolactone oxidase. Here the problem is this is present only in birds, animals like that only, not for human beings. That is why we are not able to synthesize ascorbic acid, because we are having the deficiency of L-Gulonate oxidase enzymes, right? Human beings and primates. This is very, very important. Because of the blockage of the enzyme, we are not able to synthesize ascorbic acid. That is why we should take vitamin C through our food items only. Okay. Next, what are all the MCQs or theory points we have to write? First one is, what is the site of this pathway? Usually liver. What are all the products you will get in this pathway? First one is glucuronic acid, second one is vitamin C, third one is pentose sugar. This xylulose 5-phosphate is pentose sugar. Here there is no ATP generation. Okay. And what are all the precursors of this vitamin? What is a precursor? Only one precursor, that is L-Gulonate. The enzyme name is L-Gulonate oxidase, which is absent in human beings. So these are all the important MCQ points. Now coming to the functions. What are all the functions of this glucuronic acid is? We have only two functions. One is synthesis of some compounds. Second one is xenobiotics or detoxification or conjugation reactions. Okay. The first one is synthesis of GAG, that is glycosaminoglycans, glycoproteins and glycogen. These three are the products derived from uronic acid pathway. Next one is conjugation. They always go with the glucuronic acid and makes the conjugation for the excretion of or detoxification of bilirubin, steroids and detoxification of some drugs. Very, very important function. Detoxification of drugs, conjugation of steroids, conjugation of bilirubin are the important functions of glucuronic acid. Right? Final point is, we have some drugs also to induce the uronic acid pathway to produce more amount of glucuronic acid for conjugation. One example is barbiturates. It is one of the anticonvulsant drugs. This increases the pathway of uronic acid. Okay.